So now on to chapter 9.1, where we're going to talk about glucose transport. So some key transporters that we need to know for the MCAT. We're going to say GLUT2, and you need to know this is found in the liver and in the pancreas beta cells, okay? You need to know that these have a high Km. They in turn act as a glucose sensor. So next we have GLUT4. GLUT4 is found in your adipose tissues and your muscle tissues, and these have a low Km. Insulin increases uptake by increasing transporter expression at the membrane. So next, let's go over glycolysis. Glycolysis occurs in the cytoplasm. It could be anaerobic or aerobic, and we all know it converts glucose into 2-pyruvate, 2-ATP, and 2-NADH. So some very important enzyme in glycolysis, we're going to start with hexokinase, or you may see it as glucokinase. So it phosphorylates glucose, okay? Meaning we're going to add a phosphate to glucose, and we're going to turn glucose into G6P. So the 6 tells us we added the phosphate on the 6 carbon. Next we have, we're going to keep going with hexokinase. So it's in all tissues that has a low Km. And glucokinase is in liver/pancreas and has a high Km. So PFK1 is one of your rate limiting steps, and it's activated by AMP. AMP meaning adenosine monophosphate, so it's only attached to, there's only one phosphate attached. It's also activated by F26BP, and it's also activated, and then it's inhibited by ATP and citrate. Okay, and this is something you should definitely know, and hint, hint. So next we have glyceraldehyde 3-phosphate dehydrogenase, and this is where you form NADH, okay? Next we have PGK. So, and we also have pyruvate kinase, okay? So phosphoglycerate kinase and pyruvate kinase. You may just see this as PGK and then PK if you're on like an exam. So this is substrate level phosphorylation, meaning this is where we generate our ATP. So next we have fermentation. So fermentation, the reason why it happens, the goal is to regenerate NAD+. Okay, so fermentation happens when you have too much NADH. So if you have high NADH, you're going to go into fermentation because you would like to regenerate NAD+. Okay, anaerobic glycolysis leads to lactic acid fermentation. So some more important intermediates, DAP is used in triglyceride synthesis, and then 1,3-BPG and PEP generate ATP, okay? Definitely important to know, and they generate ATP via substrate level phosphorylation.